Hey, what's up everyone? I wanted to talk to you guys a little bit about genomes, and I'm going to split this again into two kind of shorter lectures where we talk about what genomes are, what genomics is, and why it actually matters. Let's start with this term genome. So if you've seen that term before, usually the word gene is in there. Usually we're thinking of genes. But a genome means a very specific thing. A genome means all of the genes that you would find in a particular organism. Now, this particular topic for us is going to be very human heavy. We're going to talk about the Human Genome Project as part of this. But we do genomics on other organisms, and I'm going to mention that as we go through. So when we're doing this study of genes, which we call genomics, what we're doing is we are looking at the location and trying to study the function of the genes that you'd find in an organism. So we're looking at the entirety, all of the genes. How do they function individually? How do they function together? And here's some of the stuff that we know, just to throw out a few numbers. So for example, there are about 25,000 genes in the human genome. We had actually estimated that there may have been as many as 100,000 genes. So this was a bit of a surprise once we actually made this discovery. We have looked at the genomes of other organisms. So for example, fruit flies. Now fruit flies are really commonly used in genetic studies. We'll talk a little bit more about that later on. But those genomes are much smaller. They only have about 13,500 genes. There's another organism that we're going to talk about later. It's a roundworm. It's called C. elegans, which you can see here. C. elegans actually has more genes than a fruit fly. It has 19,000 genes, but is a much simpler organism. So the number of genes doesn't necessarily correspond to the complexity of an organism. And part of that has to do with the organization of the genes. So we'll talk a little bit more about that. Now, another field that's related to genomics is this field called proteomics. And if you remember, genes are linked to proteins because our genetic code makes messenger RNA. That messenger RNA then is used to build a protein. And so for human beings, for our proteomics, we have about 100,000 proteins that our genes make. And so one of the things that science is looking at now is how do we get that alternate splicing? How do we produce those alternate proteins from the same number of genes? And that's a whole other field. So I wanted to talk a little bit before we get into how we do this, sort of why. So what are the sort of things we can discover by looking at the genome? And one of the big things we can discover is actually which genes are considered to be maybe, and I hate using this term normal, but genes that allow the organism to function versus genes that are broken, and maybe we would consider them disease genes. So being able to compare those two to understand how the genetic basis of disease works, that was certainly an underlying reason for us to sequence the human genome. But we can also discover other things. By comparing two organisms, we can look at the function of a gene in one organism to figure out how it might work in another. And a really good example of this is if you look at the genes that control, for example, how we take food and turn it into energy. All living organisms have to be able to do this. But if you want to study it in a human, where you have a very complex organism with lots of cells, and indeed our cells actually work a little bit differently, depending on where you are, how their energy needs are different, that can be very different than if you're talking about a single-celled organism like a yeast. And so it's easier to manipulate that yeast cell to discover how the genes work. So we can do a comparison and that will help us determine function. Now, one of the techniques that became very, very important in this whole process is this technique called DNA sequencing. Basically what it is this. It's determining the order of nucleotides in the genome of an organism. So the idea here is you want to determine the order and location of all the adenines, guanines, cytosines, and thymines in a piece of DNA. Now, The technique to do this was developed in the 70s, it was refined in the 80s, and it's gone through a whole bunch of changes in the 90s and 2000s. But the basics of it is this. We are going to basically be doing PCR. We're going to be amplifying the DNA. So here you can see a sequence of DNA. But instead of using normal nucleotides, we do use those, but we add in some nucleotides that have a fluorescent dye on them. And then they're designed specially so that if they get added to the DNA strand, they terminate the PCR process. So in other words, if I'm trying to match up this DNA strand here, and I get one of these special nucleotides that's a terminator. So I got an adenine, and I added it here to this thymine. If I paired it with the DNA polymerase, I would automatically end that process of polymerization. So I wouldn't go any further. Now, if I had a bunch of normal nucleotides that got paired, and I got to this guanine here, and I paired a terminator cytosine to it, that DNA copying process would end there. And so what I end up getting is a bunch of different length DNA molecules. So I'll get a bunch of different strands. Now, the reason we use fluorescent dyes is we make each one of the nucleotides, A, G, C, or T, a different color. And so, for example, this particular sequence, A, T, C, G, this ends in G, this one ends in G, this one ends in G. They're all different lengths, but they all end in G, and they'd all have the same color G to them. These all end in C, these two end in A, these end in T. So I would then do gel electrophoresis. So you run them out on a gel, and I would do each of these in a separate tube. So each fragment that I get here in the tube that has the terminator guanine in it, this would tell me that each of these fragments ended in G. And when I read this gel, so this is the smallest band, this tells me that the first nucleotide here is A, and because this is the T one, this one ends in T. And you can do this process, and I actually did some of this as an undergraduate. You can actually figure out the order of nucleotides. Now this takes a very long time, and now we have automated systems. And so we use fluorescent dyes, and we actually have a laser that shines off of them. We do gel electrophoresis, but we do it in a microcapillary tube. It's a very small tube, and the laser shines on these fragments as they pass through the gel, and then they're picked up by a detector, and then a computer actually reads them. And so this process that I used to do, it would take 2 weeks to read maybe several hundred nucleotides. Now we can read several thousand nucleotides within an hour or two. So this process of sequencing is sort of the basis of how we understand genomics, and it's going to be the basis of the stuff that we talk about from here on out.